Okay, so good afternoon, everybody. Because it's past midday, correct? I apologize for the delay. I'm actually on call this week, and we cannot plan things very well. But we're very happy to have here Dr. Antonio Ieva. He is certainly one of the best speakers that we can have for this lecture that he's going to give to us. He is a neurosurgeon from Italy. He is a professor of anatomy at the Medical University of Vienna. He has a PhD in clinical neuroscience. I had to write everything because there's so many titles. He was a fellow here at St. Mike's.
Still now.
Still now. His research and interested in computer clinical neurosciences. So he correlates computer advancements with neurosciences. And he will speak to us today, and I think everybody will learn a lot from his lecture. And he will have a time for questions at the end.
Right.
Correct?
Correct.
And thank you very much.
Thank you very much for this very kind presentation.
Thank you.
And I would like to thank also Dr. Ore Rothstein for this kind invitation to this popular science lecture, because I'm really very proud of that. As told, my name is Antonio Ieva, and I'm a neurosurgeon working here in skull-based surgery. So it's a clinical fellowship working and dealing with the brain tumors and the tumors of the skull base. Do you have an idea what is a skull base? Well, me neither. I'm trying to just to figure out what I'm doing. But anyway, it's a kind of a transitional surgery between different competences, between ENT, craniomaxillofacial surgery, orbital surgery, and the brain surgery. By the way, as I said, I'm working in a research field in what we call computational clinical neuroscience. What does it mean? The application of computer science to medicine. In my field, especially to neurosciences and to the study of the brain. So fractal geometry, which is the topic of today, is the main topic, is one of the models we are applying for these computational studies. I'm very proud to put this presentation, this picture, because this is the bridge between, St. Michael Hospital and Li Ka-shing, just to show that in this hospital, as many times in several places in the world, it's enough just to cross a bridge to cross the different competences. Because basically, I'm a surgeon, and in the Middle Age, surgeons were just barbers. But now we don't do barb anymore, something more. And we don't just cut and put stitches, but we do also researches. And this is a fantastic hospital where you can really just jump the bridge to get collaboration for scientific research. So if there are students or future researchers here, or actually researchers, this is a very good place where you can work. And this, what I'm saying, is shown also by the symbol of the university, the Department of Neurosurgery of the University of Toronto. There is a DNA with the neuron and the brain. So this is the connection, the very tight connection between research, basic research, clinical research, and surgery. So well, let's jump to my topics, fractals in neurosciences. We are talking about a very complex topic. We are talking about complexity, but believe me, the most complex thing you have to deal with is my accent. So when you get my accent, it's 50% of the job is done, and everything will be much easier. So after that, what is basically fractal geometry? We start with these very popular science slides, because basically it's the geometry of the complexity of the natural objects. We have clouds, we have trees, we have rivers, and all these objects, they present a kind of geometrical complexity which cannot be quantified by means of simple rules and models, as we will see. This was understood by Benoit Mandelbrot, basically a mathematician, but he was a genius introducing in science the fractal geometry of nature, just noting that natural objects are too much complex to be studied by means of the normal geometry. Which one is the more normal geometry? The one that we study in the school, Euclidean geometry, as we'll see now. So after the introduction of his book, which is the milestone of fractal geometry of the nature, there was an explosion of the application of this mathematical model to physics, geology, economics, And in the last 20 years, also medicine, biomedical applications, and the biggest explosion is a little bit in computer graphic, because as you can see, the fractals have very, very nice patterns. You can put a lot of colors, very nice shapes, and many times you don't know, but you are looking television, movies, cartoons, comics, which are fractal algorithm based, like the Pixar, the famous Pixar of Steve Jobs. They made a lot of movies with the fractal based algorithms. So again, let's see some natural objects. The lightning does not fall down like a straight line, as Mandelbrot was used to say. And we have other objects, natural or also biological objects, like the bronchial trees, which have a complexity that in one word can be said that they have a roughness. This roughness can be quantified by means of this mathematical model. So what is a fractal? Let's use Wikipedia, which is our encyclopedia right now. And we find the definition that, well, is not really that easy. A fractal is a mathematical set that has a fractal dimension that usually exceeds its topological dimension and may fall between the integers. Fractals are typical self-similar patterns, where self-similar patterns means that they are the same from the near as far as from far. Very easy definition, right? You understood everything. So let's test myself. As Albert Einstein was saying, if you cannot explain it simply, you don't understand it well enough, right? So this is a test for me if I understood well enough to explain you. So fractal dimension. Dimension is a parameter to quantify something. In this case, the geometrical complexity of the objects. Fractal means a number which is not integer number, is fractal, from Latin fractus, which means not integer. So what is the topological dimension? In the Euclidean geometry, we use the three dimensions, the length, the width, and the height. So, and we have the point, which is represented by the number 0, topological dimension of the point 0. The line is 1. The plane is a 2, that's why we say the two-dimensional object is a plane. And the three-dimensional object can be a cube, where there are represented all the three dimensions. Well, the fractal dimension is a number between the topological dimension. So it's a number between 0 and 3, not integer number, giving you the quantification of how that object fills the space in which is embedded. For example, this line, how fills this plane? 1, because it's a single line. But if we have a different shape, the fractal dimension is increasing because it fills more the space in which is embedded. And if we have an object which fills almost all the plane, the number can be 1.99 because it's a much more geometrical complex. If we want to do another example, now we are 50% filling all the space of this room. So our two-dimensional room is almost completely filled by us. So the fractal dimension of us in this room is very high, is close to 2. If we were like 10 persons just there and 2 persons there, the fractal dimension were different because the pattern is different. So although we can be always 50 persons, the fractal dimension can change according to the pattern how we distribute ourselves. If we put every one of us going in that corner, the fractal dimension, the density of us is always the same. So the Euclidean geometry is the same, but the fractal dimension is changing because it's changing the pattern of the distribution. So Mandelbrot introduced this concept in a landmark article published in Science in 1967. Basically, in this article, talking about the coastline of the Great Britain, is introducing the two main concepts, the fractal dimension that I told you, and the statistical self-similarity. The question of Mandelbrot was, how long is the coastline of Great Britain? He was studying the atlas of the last years, and he saw that the coastline of Great Britain was increasing, atlas by atlas. So was really the Great Britain becoming bigger? No, was the resolution of the ruler becoming bigger. So if you consider this length, but you consider that you don't do the coastline with a car anymore, but you do by feet, the resolution is higher because you are able to follow more the roughness of the coastline. So at the end, it seems that the length is going higher and higher, but it's not true, obviously. So what is that is self-repeating over time? The same pattern, which is called self-similar pattern. So he applied just an algorithm to to quantify how geometrical complex is the roughness of the coastline. He gave a number, and this number is a quantitative number which can be easily compared. Because, for example, we have a number for Great Britain that basically for sure is lower than the coastline of the Norway, for example. Because Norway has a lot of fjords. So the geometrical complexity of this coastline is much higher. And this is the concept of self-similarity. It takes some seconds, but I like so much from YouTube to present this small video because it's a fantastic video to show the self-similarity concept. Basically, you find this is the Mandelbrot set. So it's a mathematical set that how long you can magnify doing a zoom, you find always the same pattern. Wherever you go, you find the same pattern. Sometimes the shape changes a little bit. So we say that the Mandelbrot set is the generator of all the fractal set. And the moreover you get a magnification, again you find different fractal patterns until you range a very, very high magnification, where at the end, what do you see? The Mandelbrot set again. So it's a mathematical concept. From minus infinity to plus infinity, we have always the same pattern. So from the atoms to the universe, we have always the repetition of the self-similar pattern. And this is the concept of self-similarity, which is a true concept in mathematics. But in biology, what we can say about biology? Well, a lot of patterns are self-similar, but not from minus infinity to plus infinity, just in a range of magnification, what we call the window of self-similarity. And we'll explain this. So just to summarize again, the Mandelbrot set has this shape, and if you magnify 6, 36, 8,500 until 2 million times, you find always the same patterns. It's obvious that the computer graphic can help us to identify the patterns and to give colors and to give rise to beautiful fractal representation of the mathematical set. So let's come back to the biological structure in nature. A lot of biological structures have a self-similar pattern. If you cut this structure, you find always that there is a self-similar pattern repeating in different orders of zoom. And this is the dream of every neurosurgeon, a self-similar hand. You find always smaller and smaller fingers. Obviously, this is just I think that there is a presentation for surgeon for a fractal pattern. But this is a best example for you to reach the topic I wanted to deal with. We have two trees, two different trees in terms of shape. This is, everyone knows, a weeping willow, and this is a pine. You have not to be a botanist or expert in these kind of things to understand there are different trees. So it's like that just looking at the shape, we are able to identify what's the name of this tree. So let's say that the shape of the tree is giving you information about what is really the tree is. So it's like that the tree, in terms of shape, is a kind of fingerprinting, just in terms of shape. So how we can quantify and describe this shape of these trees? We can apply Euclidean geometry. So again, it's the geometry we study in the school. Simple rules applied to Platonic objects. Platonic objects are perfect objects existing in mathematics. And also this table is a Platonic object, but in nature does not exist a so perfect structure, because nature is much rougher. So Euclidean geometry fails in the description of the trees, because those two trees can have exactly the three same dimensions. Maybe they are same height, same width, same length, but we know very well that they are different objects. And we are still able to quantify and to say that they are different. So what is the difference? The shape again. And we can say that the fractal dimension is different because they have a completely different conformation and geometrical shape, and they have a different capacity to fill the space. By means we can say, just looking 1 second, that there are two different trees. So the tree is a very nice mathematical model, as you can see here. This is the mathematical tree of the representation of the self-similarity, where there is the arborization system, where you find always the same pattern, but smaller and smaller. And the arborization systems you find a little bit everywhere, also in the human body. the bronchial tree. The brain itself is a fractal structure. The sutures on the skull are not a straight line, but are very rough structures. And you cannot quantify these by means of Euclidean geometry. By means of a fractal dimension, yes. And also the time series. When you do the beat of the heart, the electrocardiography, if you see the wave, it's not a straight line, because you are dead if it's a straight line. But it's a very rough line, which can be quantified by means of a time series analysis, which is, for example, fractal analysis. ECG, so the study of the brain waves, the same. And again, the vascularization of the brain. This is a cast of the arteries in the brain. I prepared in the laboratory of anatomy in Vienna. This is a bronchial tree structure, very similar arborization. And this is the neurons, how they do arborization and network to connect each other. And again, very nice example, the bronchial tree and the botanic tree. They have a very similar pattern. It's a little bit like the nature finds some rules, some cheap rules, and applies these rules to many different things in nature. So why I'm talking always about arborization and tree? Do we have only trees in mind? Yes, why not? If you see the brain from an anatomical point of view, it has a lot of arborization. The white matter of the brain and of the cerebellum are very well arborized. If you see the white matter of the cerebellum, in the old book of anatomy, you find this definition, arbor vitae, which later means the tree of life. So it's a tree. And again, the brain self and the vascular structure of the brain in physiologic and pathological condition. So the artery of the brain in a normal brain, and where is a malformation called the arteriovenous malformation, which is basically a tangle of vessels. And the fractal dimension can be applied for the study of these objects. Again, the neurons, they have different shapes. Maybe they have the same quantity in terms of number of dendrites, but maybe the geometrical complexity, as you can see from the shape, is completely different. And these can be quantified by means of the fractal dimension. So this is one of the applications I would like to talk, and it's one of my research. So the brain has a very complex microvascular system, but this system changes when there is a tumor. It's like the tumor is a different tree. Goes there and makes, by means of a process called angiogenesis, creates new vessels and creates new patterns of vessels. This is very well known since many years. And why the importance of the microvessels? Already in 1972, there was the idea to quantify the vessels in the histological specimens of a brain tumor. There was the idea that maybe these vessels can be a morphometric biomarker, so they can help the pathologist to say what kind of a tumor is. Is highly vascularized, is low vascularized, is quite relevant, especially today because we know the importance, thanks to Folkman, we know the importance of the process of angiogenesis, so the creation of new vessels for carrying blood into the tumor. And we also use in neuro-oncology some medications, some drugs working on the vessels, trying to cut off the vessels. So anti-angiogenic therapies, which is very relevant for patients, obviously. But the problem is that about the microvessels, we know a lot of information from a genetic and a biological point of view. But still now, there is still the lack of a gold standard to quantify the microvessels. So the first thing I did in this research some years ago was to study what the other people in the literature they did, which are the parameters used to quantify the microvessels. So I differentiate a box Euclidean parameters. So now you know Euclidean geometry, we are just counting how long are the vessels, how many vessels, the density, the size, the total vascular area. But there are also more complex parameters, the fractal parameters, quantifying the geometrical complexity of the vessels, and the other parameters like the compactness, the shape factor, etc. Each of these parameters has a pros and cons. The most used one in literature is the microvessel density, which is quite a good parameter. They published a lot. Still now they are publishing a lot on this parameter because they thought that it has a prognostic value. The problem is that the MVD, you count just the number of the vessel in the so-called hot spot. So the pathologist chooses a spot of the histological specimen where he thinks that there are more vessels. On this hot spot makes the manual or computer-aided quantification of how many vessels are there. What's the problem? The hot spot is a very subjective chosen. Different pathologists can choose different hot spot. And at the end, the number you get is statistically not comparable anymore because there is a very high inter-observer disagreement. So you can get different numbers on the same histological specimen of tumors, and this cannot be a good morphometric biomarker because you cannot do comparison. So why we don't use the fractal dimension for quantifying the microvessels? Maybe the fractal dimension, as I told you, this number between 0 and 3, can quantify and trying to describe what I called the microvascular fingerprinting. So if two trees are different, have two different shapes, and you can say that they are different trees, why we don't try with the tumors? They have different microvascular patterns. We quantify the fractal dimension, and we see if the fractal dimension of this microvascular network is able to give us some information about the nature of the tumor. What kind of tumor is benign, cancer, what kind of cancer, so subtypes. So the application of the fractal dimensional model to the vascular trees model. And you can do into 2 dimensions when you have a slide, a specimen, histological specimen of the tumor, or in 3D when you have a reconstruction. The reconstruction can be done in the electron microscopy or in neuroradiological imaging like an MRI. So to test this model, we started with the application of fractal analysis to a very simple model. A simple model for me was the pituitary adenoma. In the brain, there is a very small gland, but which is very important. It is the master gland because it controls all the hormones of the body, the pituitary gland. The pituitary gland has a transformation in benign tumor called pituitary adenoma. We know very well that the pituitary gland is more vascularized than the pituitary adenoma. Can we quantify that? So we applied a model in which we use some markers to identify just the vessel. This is an antibody called CD34. So by means of immunohistochemistry, we colorate by a brown coloration all the vessels. And we show that the pituitary gland is not just more vascularized than pituitary adenoma, but that the fractal dimension is higher. So this is the first demonstration that the pituitary gland is not just more vascularized than the tumor, but is also more geometrically complex than the tumor itself. And the other idea was creating the vascular catalog, extracting all the vessels to see also the variability in terms of shape. So the simple model could be translated to something, let's say, more relevant from a clinical point of view. This is the Glioblastoma Multiforme, GBM. It's the most malignant kind of cancer existing because this is a kind of brain cancer. We can do, the best we can do is surgery, resection of the tumor, radiation, chemotherapy, but these patients still die after an average of 2 years. So, and this is a tumor which is classified in the classification system of brain tumors always as a 4th grade, the most malignant tumor, but is always a 4th grade tumor. And the problem is that this 4th grade tumor, which for the pathologist is very homogeneous, 4th grade, at the end is a very heterogeneous. Because if you see the vessels, and that's why it's called a multiforme. Multiforme means multi-shape, from Latin multiformis. And it means that it shows many geometrical patterns in terms of everything, and also in terms of microvessels. Here you can see the same tumor, not so many vessels, a lot of vessels with a homogeneous distribution, and a lot of vessels but with a very specific pattern. Here we have a necrotic area, and we have some kind of palisades. The pathologists call these garlands-like distribution of the vessels. So the pathologists, they know that the same tumor have different patterns, but they are not able to quantify this. They just describe, okay, garland-like structure. But this is very relevant because it was shown that these tumors, these 3 different tumors, they respond in a different way to the chemotherapy, to radiations, etc. So it's relevant from a clinical point of view, although we still call it glioblastoma multiforme 4th grade. So this is a project I did when I was doing a PhD in Vienna, in Austria. And I show you how I did it just because for whoever you want to do research in the future, maybe helps a little bit to track a plane. I did 2 frames of the project to show that really the tumor have a microvascular fingerprinting. The neuropathology frame and the neuroradiology. So dealing with the histopathological specimen of brain tumors, dealing with MRI images. 2 different phases. One methodological, so collection of the cases, technical hits, image analysis, fractal analysis. You can figure out that in this phase there are a lot of collaboration with computer scientists and engineers. Thanks God, my best friend Carlo Russo in Milan is a genius computer scientist, helped me in all this phase. But after you standardize the computer-aided model, you have to try to translate all these computational things into clinical applications. So, clinical step, I'm a clinician basically. I'm a neurosurgeon, so I deal with the patients with the brain tumors. So I have to see if this can be translated in this clinical application and the morphometric biomarker giving to the pathologists, to the neurosurgeons, to the clinicians, some useful information. So first step, this is the virtual microscope. All the pathologists use a normal microscope. They see the specimen, they quantify the MVD in the subjective chosen hotspot, and that's it. Now we have the virtual microscope in which you can do an acquisition, a digitalization of all the specimen with a very high resolution. And you use the computer like a microscope, because just with the mouse you can zoom, and instead of change the lens in the microscope, you just zoom with your mouse. And you can get very high resolution, looking the cells, the nucleus of the cells. In this case, we see the CD34, so the endothelium, meaning we see the vessels of the tumor. Okay? So the first thing was, let's try to use not the hotspot, but all the specimen we have. The computer digitalized the specimen, and we use whatever we have, whatever the neurosurgeon is sending to the lab. We standardized an algorithm. So the idea was, let's do immunohistochemistry. Let's extract the vessels, microvessel selection, by means of RGB selection. It means that we say to the computer, please extract all the red stuff, all the rest is garbage. And as you see, the histological specimen disappears, and we have just the vessels right now. It's a very simple image analysis. You can do also with the Photoshop. This can be analyzed by means of different algorithms. This is the box counting dimension. It's the same algorithm which was used by Benoit Mandelbrot for the description of the roughness of the coastline of Great Britain. In this case, when we use this box counting dimension to the microvessels, we get a number which I called microvascular fractal dimension, which in one single parameter quantifies the number of the vessels, but not just that, also the variability of the vessels in terms of shape and magnitude, and the distribution pattern, which is the most important thing. So are we able to quantify the different patterns by means of the fractal dimension? As I show with the pituitary adenoma, yes. But let's come back to the glioblastoma multiforme. So it's a multiforme in terms of shape. Let's see if it's a multiforme in terms of microvessels. Here we have the three different patterns I showed you before. The catalogs, you see already that are different. More microvessels here, more heterogeneity, more different shapes, different patterns of distribution, and the number of the fractal dimension are different. Low fractal dimension, intermediate, very high fractal dimension. This tumor could be more malignant than this and has a completely different microvascular fingerprinting because it has a specific pattern of vascularization. And so Mandelbrot was used to say, to see is to believe. So now we can qualitatively describe the tumors. So they are different. We know it. But we can also quantify this geometrical heterogeneity. And so we quantify that the glioblastoma is a multiforme, multi-shapes, also in terms of microvascularization. So now we have a tool. This is the computer-aided analysis. We have a tool saying that this tumor, this different histological specimen, have a different distribution of the vessels with a different number. We have a pituitary gland, normal organ. We have a GBM, glioblastoma multiforme. We have another glioblastoma multiforme with a different shape, different numbers. We have a normal pituitary gland. We have a pituitary adenoma. Everyone has a number. How can we use this number? Cui prodest, in Latin said, has benefited to who? Or in American, better, who cares of this number, right? And now I tell you who cares. Putting a very large sample size of a glioblastoma multiforme, we saw that there are tumors with a very low heterogeneity, very low fractal dimension, and a very high fractal dimension. We quantified how is the coefficient of variability, how much they change this number. And we quantified that it was 44%. What does it mean? That these numbers is very different. It goes from a very low number close to 1 to a very high number close to 2. If you take a pituitary adenoma, which is a benign tumor, this coefficient of variability is just less than 10%. It means that that tumor is a very homogeneous. This tumor is a very chaotic. So we introduce a new word. There is a very chaotic complexity. And this can be quantified by means of the microvascular fractal dimension and the coefficient of variability. Other point, Glioblastoma multiforme is so multiforme that sometimes we find just a necrosis, area of a necrosis. So it makes no sense to quantify also vessels where there are no vessels because there is dead tissue. So the idea of the hot spot of the microvessel density was not that bad, basically. The problem is that it is chosen by the pathologist. So how can we choose a hot spot but reducing the inter-observer variability? Let's ask to the computer to choose the hot spot. So we introduced a new algorithm in which the computer makes a grid of standardized 1 millimeter squared boxes. On each of these boxes makes a measure of the vascular area. Where the vascular area is the highest number, this is the hot spot. So automatically the computer extracts the higher hot spot area, and on this hot spot we do the boxing counting dimension. So we don't have anymore the fractal dimension of the specimen, but we have the local box counting dimension, which is a fractal-based parameter. I don't want to do it very more difficult than this, but so this is the extraction of the hot spot. We can also put colors. So the computer just extracts the color where there are more vessels. And from this vascular area map, you can also quantify the fractal dimension on each single box, and you can do a kind of a pattern to quantify the different distribution of the vessels. We did a principal component analysis, which is a very powerful statistical tool, to see that the fractal-based parameters are the best parameters to quantify the macrovascular distribution. And how can we use this? We have different tumors. So the classification of the brain tumors is in 4 grades. The first grade, the second grade, the third grade is already malignant tumor, is cancer basically, and the fourth grade is the worst one, the glioblastoma multiforme. So here we have tumors, the second, third, and fourth grade, just in a chaotic distribution. Let's see if the computer is able to classify this tumor just according to the macrovascular patterns. So let's say put a graph where the fractal dimension parameters are increasing. So this is the line, the arrow, where the geometrical complexity is increasing. And let's ask the computer to quantify and to put order in this chaotic distribution. This is what happens. Box counting dimension. Each tumor now is put in the spot where is a lower fractal dimension, intermediate, and a very high fractal dimension of the microvessels. Not for coincidence, when we did the double-blind study and analysis with the pathologists, we found that the low fractal dimension is what the pathologist calls no clusters of vessels. Clusters of vessels and very prominent clusters of vessels. So the computer is saying something that really is in agreement with what the pathologist sees. Moreover, this tumor, in 86% of the cases, so a very high sensitivity, these tumors are second grade, these tumors are third grade, these tumors are fourth grade. So for the first time, it's possible that the computer is giving us a parameter to helping us in the differentiation of the grade of the subtype of the tumor. So we are trying to cancel not the pathologists as they think, because they think that I'm trying to replace them with the computer. No, but to helping them, giving them in their armamentarium a new morphometric computer-aided parameter, which can just simply help them in the classification. Okay? The microvessels right now are not used in the World Health Classification System to classify tumor second, third, or fourth grade, just the fourth grade, but not this grade. So this is also the first demonstration that the vessels are different, have different patterns when the tumor is more malignant or is going towards cancer. Which are the limits of neuropathology? Basically, neuropathology gives us a snapshot of a situation. It's like we have a tumor, we take a piece of the tumor, we do a picture in that time, and we do a lot of analysis. The tumor is a dynamical process in a super dynamic person, which changes day by day, right? But we are not able to follow up this person and this tumor anymore because we have the snapshot forever. So the dream of the future is that we can follow in vivo over time the tumor. This is our dream in the future. This is the brain of a rat, of a mouse, with the glioblastoma multiforme implanted there. And these guys in Nature published that the fractal dimension of this is very different from this, as you can already see. It's completely different geometrical patterns. So they quantified, but you can already see that the shapes are different, completely different shapes like the two trees. And this has a different geometrical complexity of the vessels. Well, we cannot do this on patients. We cannot open the head and putting two double photons electron microscopy. And also because the penetration of the microscope is just 2 millimeters. the brain of the humans are much bigger than the brain of the rats. So we cannot use it in vivo right now, this technique. But what we can use in vivo on patients is radiology. So MRI, we can do MRI without any problems. No side effects like CT scan. We don't have radiations. So we can follow up very well patients and the tumors by means of MRI. Fractal analysis has been applied quite successfully in many, many subfields of neuroradiology. I worked on some specific sequences in MRI. The name is SWI. They give rise to the identification of vessels, especially veins, and the iron deposits, like blood, microbleedings. And in Vienna, we had also the luck to have the ultra-high field magnetic resonance. What does it mean? Magnetic resonance use a magnetic field. In general, in all the world, they use 1.5 Tesla. It's a quite strong field. But many hospitals are switching to 3 Tesla. This hospital has the 3 Tesla as well. 7 Tesla is the highest power we can, highest magnetic field we can use on humans right now. And for example, in Toronto, there is no 7 Tesla right now in for clinical application. In the world, there are some 7 Tesla MRI, but they are more used for research. But still, you can use it for brain tumors. And so we started the experience. This is something I published on Neuroimage, just to show that what we could see in the anatomical dissection of the brain could be also seen in 7 Tesla MRI, because the resolution is much higher. We can see submillimetric veins, vascular networks, and we can also reach almost histological definition. So for the first time, we saw a submillimetric vein, which was never seen in vivo, but just in vitro, just on the cadaver. So this comparison got the cover of this top journal in neuroimaging. Thanks to this experience, we used the 7 Tesla and the SWI together for the quantification of the brain tumors. This is a recurrence of GBM. As you can see here, if you do a 3 Tesla MRI, sometimes you have just 2 colors, but you don't see a lot of a signal inside the tumor, within the tumor. But with the 7 Tesla, we see a lot of things. As you can see, we are still very far from the resolution you saw before with the microvessels, because this was histology. Here we are in vivo, we are not anymore with the microscope. But still is a very high advancement, because right now we can see what could be microvascular network. So these small tubes could be small vessels. And we see these dots, which are basically deposits of iron, meaning blood, microbleeding in the tumor. So the radiologist just says, okay, this patient is undergoing a 7 Tesla MRI every 2 weeks. You see that there is a recurrence of the tumor, and something is increasing. There are more dots, much more dots. But it's just saying that every 2 weeks the dots are increasing in terms maybe of the number. So you can quantify the number maybe, but in general they don't do. So the idea was, let's apply the same model of the fractal dimension to quantify the geometrical complexity of the SWI in the tumor. So we applied the model. We did the algorithm, which is very similar to the one I showed you before for the histological specimens, with the difference. Right now, we can use not just one slide of the brain, but all the slide of the tumors. So a 3D reconstruction of the tumor, because the MRI gives us a multiplanar reconstruction of the tumor. So this time we have a number which is between 0 and 3, because it's a 3-dimensional object. And the reconstruction in 3D shows that the tumor is increasing in terms of SWI, but also the fractal dimension is increasing. So this is a patient that every 2 weeks has more vessels in the tumors. And this is the first quantitative objective parameter to follow up patients over time, because we can see patients how they are going over time, but we can also follow up a medication. Remember the medication I told you about before, the bevacizumab, is an anti-angiogenic treatment. So we give this medication to the patient. We hope that the vessels, that we stop the angiogenesis, and maybe we try to starve the tumor. And we can follow up the function of the bevacizumab is working or is not working, just to see if there are more macrovessels over time, and if the fractal dimension is changing, is increasing or decreasing. So we did exactly what we did before with the different grades of a tumor in histology. We did also in radiology. Let's take some patients with a second-grade tumor. We already know that histologically are second-grade, third-grade, and fourth-grade. And let's see if the computer gives us a number which quantifies and classifies the tumor as the radiologists and pathologists do. And again, we found a very nice statistical agreement between lower fractal dimension of the SWI pattern, second grade, intermediate, third grade, and the very high geometrical complexity of the dots, very high fractal dimension. This tumor is a glioblastoma multiforme. So again, the computer is helping us with a morphometric biomarker. So the conclusion of this part of the project is that the fractal analysis is a valid tool to quantify in an objective way different microvascular networks. It's able to discriminate different types and subtypes of brain tumors in neuropathology as well in neuroradiology. So basically, it's the model to quantify the hypothesis of the microvascular fingerprinting. And maybe it's also able to help us in the follow-up of the patients in vivo, not in vitro anymore. So that's why I put this picture. Similarity between the vessels of the brain, similarity with the trees, botanic trees, but you can apply the same model for this quantification of this arborization to the quantification of the arborization of brain tumors in neuroradiology as well in neuropathology. And so the importance of the fractal-based morphometric what is? Is the science of quantification of the objects. Is that the computer-aided and fractal-based morphometric analysis of microvascularity and also other biological objects is first of all objective. There is no choice of the pathologist or radiologist. The computer does use it by means of using the same algorithm every time. It's realistic because it's very close to the reality. It's very close to the fractal geometry of nature, not to the Platonic object, which does not exist. It's reproducible because if I do with this computer with the same algorithm here in Toronto, everyone in the world can apply the same algorithm. And so when you get a number, you can compare from a statistical point of view. So you are quantifying, not anymore comparing, not anymore apples and oranges. You are quantifying and comparing the same object. And basically, this parameter is a potential prognostic and a predictive biomarker. So the robot, the computer, is not superior to human mind, but it can help a lot in quantification of different objects for what we call as the descriptive analysis of different patterns. So these results could be translated to clinical practice to help physicians in discriminating. This is a little bit to summarize. Different pathologies. For example, malignant cancer versus benign tumor. Different states of the physiopathological condition. So neoplastic versus non-neoplastic. Normal brain versus brain tumor, or whatever it is, which is not a normal brain. Temporal changes in the same pathology followed over time. For example, we have the same tumor, but we can follow up over time the changes of the signal inside the tumor. And we can also quantify the morphological variations in the anatomical structures. The limits of a fractal analysis. Basically, there are a lot of limits. It's a mathematical model. So we need hours and a lot of experience to talk about the limits of fractal analysis. So to summarize in a popular science lecture, I like to use a sentence of Heisenberg, the father of quantistic physics. What we observe is not nature itself, but nature exposed to our method of questioning. So basically, we are not seeing really as the nature is, but we are looking at the nature with a microscope, with a virtual microscope, with the MRI. The MRI at the end does not give rise to the identification of vessels, but are pixels. So we are playing with pixels basically. So at the end, we are just working with something quantifying something else. But let's remember this is a model, so can be wrong the MRI, can be wrong the histopathology, can be wrong the immunohistochemistry. So we have just to try to standardize, and the fractal analysis is a model too. So it's a mathematical model with the pros and the cons, obviously. And there are plenty of pros, as you saw. So whenever you hope that is finished, no, this is just the beginning, because this was the part of the application of a specific part of clinical neurosciences, brain tumors and microvessels. But I translated whatever I was doing in Europe, in Italy, and Austria, here, to more clinical application. So that's why I put this picture, because it's my bridge between when I arrived here in Toronto. where I was trying to quantify and applying these models also to clinical application. As you can see here, two different models that now we can summarize quite quickly. For example, look here. It's not very sharp because this is a very old slide, original slide from Mandelbrot. And Mandelbrot was talking about these waves, how they change over time. Specifically, he was talking about how the price of a cotton is changing over the last 100 years. And he saw that there is a self-similar pattern. Every 10, 20 years, there was a different pattern which was repeating after some years. So he identified that the prices were following a kind of self-similar pattern. And this was, so the financial prices, increments, and the models was quantified by means of a fractal dimension and fractal geometry. And there are two books that you can find in Amazon, written by Mandelbrot, in which he applies the fractal and scaling in finance. And who studies economics, he knows these kind of models as well. The point is that when I saw this, I read this book and I saw these waves, in physiology, neurophysiology, and neurosurgery, we have something very, very similar, which is the wave quantifying or describing as the intracranial pressure. So when we have a patient in trauma, after a severe trauma, they are patient in coma, very sick patient, sometimes we put a transducer, we do a bar hole, and we put a transducer in the brain. And this gives us information about the pressure of the brain. If it's a too high pressure, maybe we have to do something, a medication or operations. And if you do a representation of these waves, you find something which basically was a little bit similar to this wave. And so the idea was why we don't use the intracranial pressure waveform and we quantify with the same model that Mandelbrot was using for the cotton distribution. So first we published a kind of review. Is it feasible? Do we have the tools, the mathematical tools to do that? The answer is yes. So self-similar pattern exists in the heart as well in the brain. And the box counting dimension and the different algorithms can be used to quantify and describe the different patterns of the waveform over time. But why could be relevant? Because this is still a project still in progress. We want to see if a small pattern can differentiate or can predict how the patient is going from a clinical point of view later. Meaning that if the patient, something is happening in this part, we want to see if before, not just the single number, high blood or high brain pressure, but a single pattern can predict that something will happen in the future. So we are looking for some predictors able to give us some information for prognostication. Anatomical question, as I told you, the cranial sutures. We have this suture between the different bones of the skull. You see that they are not straight line. So how we can quantify this? The fractal dimension gives rise to the different geometrical complexity description of the sutures, and we can quantify normal anatomy, different species if they have different patterns of the sutures, or different pathologies, like in children who can have some deficit in the fusion of these sutures. These are just examples how you can extend the fractal dimensional model. So again, SWI, you know that you can see veins and the vasculature and the microbleedings. So we have a lot of trauma patients. This is one of the biggest neurotrauma centers of Canada. We get a lot of patients. We do SWI, and we see, what do we see? We see a lot of dots sometimes. Our patients awake or in coma, but many of these dots are microbleedings. So there are some dots of blood in the brain. How can you quantify this? You can count, but now you know that maybe it's not enough to count. If there are 1 dot or 10 dots, maybe clinically exactly the same. Maybe it's the geometrical complexity of where and how these dots are that is more important. So we are trying to apply this new software for fractal analysis of the geometrical complexity of the dots in the brain to give us a fractal dimension of this pattern and to see if this is a biomarker to help us in prognostication. So different patients with the different patterns of bleeding and trying to quantify the fractal dimension of the SWI patterns related to the clinical outcome. We are trying to extend what I already did with the brain tumors. Now we are studying more brain tumors, different brain tumors, not just glioma, not the tumors of the brain coming from the glia, but also meningioma, lymphoma, metastasis, and other kind of tumors. we are applying the fractal model of the microvascular fractal dimension to different kind of tumors. With Dr. Kovacs here in St. Michael Hospital, we are working on pituitary adenoma, and we are trying to use different subtypes of adenoma and different types of immunohistochemistry, CD34, CD105, which identifies the neoangiogenetic vessels. And another model where we submitted now to journal is the arteriovenous malformations are kind of a tangle of vessels in the brain. Patients affected by AVM are born with an AVM. They can live all the life without any problems, but in the same time they can have seizures or they can have bleeding. This bleeding can be also very severe and they can die. So it's very important to try to understand how quantify and describe the AVM. And basically, many people, they do just how many vessels or what's the size of the AVM. So I just thought, okay, let's apply the fractal dimensional model also to the AVM. And so now we are doing exactly the same to see if the AVM has a different geometrical pattern and if this pattern can be useful. And we already shown that it's useful because it's a predictor of the patient undergoing to gamma knife radiosurgery to see the patient who will be responder to the treatment versus the patient who will be not responder. So basically what I'm working on right now in Toronto, in St. Michael Hospital, are different projects, AVM, intracranial pressure, brain tumors, but everything is is analyzed by means of a computational fractal-based and artificial intelligence-based analysis. So there are also people from the Ryerson University. Professor Alireza Sadegian, for example, is helping me for artificial intelligence-based algorithms, just to try to help to use the same algorithms for a very different application in clinical applications. So I call fractal analysis and computational neuroscience a little bit like a Rosetta Stone for describing a different phenomena. So you need a lot of collaboration around the world. That's why I did this fractal lab, which is a fractal because it's not a physical lab, but everyone in the world can join this website and can read information or contribute by means of his expertise in computer science, informatics, etc. So different models, different things going with the same model, the description of a fractal brain model. And I want to conclude with this picture, which is in German, but I can translate it for you, because now you guys know everything about the self-similarity, so maybe this helps you to take home message. Hey, I am a broccoli, and I look like a tree. Hey, I am a nut, and I look like a brain, as you can see. Hey, I am a mushroom, and I don't like this game.